So, what is it? Codominance. Achha, first let us talk about multiple allelism. Multiple allelism. Multiple allelism. Allelism. Okay. The best example of this is ABO blood grouping. ABO blood grouping and eye color of Drosophila. Eye color of Drosophila. What is Drosophila? Fruit fly. All of us, we know that. Okay. Now, under ABO blood grouping, what we are going to study. So, there are three alleles: IA, IB, and IO. IA, IB, and IO. Three of the alleles are there. But these three alleles are present on. Three alleles are present on same locus or different locus? Same locus. Three alleles are present on same locus. Very, very important. Now, according to ABO blood grouping system, which was given by blood grouping system, ABO blood grouping system, it was given by two of the scientists named Carl Landsteiner and Wiener in the year 1940. Carl Landsteiner and Wiener in the year 1940. Now, they gave this particular concept. According to this concept that, what is it? The first one is the phenotype or the blood group. This is blood group, which is also called as phenotype. So according to ABO blood grouping system, how many blood groups are there? 4 blood groups are there: A, B, AB, and O. What are the antigens present? The next one is antigen, and these antigens are present on the membrane of RBC. They are present on the membrane of RBC. What is the membrane of RBC called? Who is going to tell me? Durba. Membrane of RBC. Donance membrane. What is the membrane of RBC called? Donance membrane. And their antigens are present on the Donance membrane. So, if the blood group is A, that means definitely the antigen present on the RBC membrane is A. So, we can call it as what? This is antigen. The next one is what? Antibody. So, when we talk about antibody, that means they are present where? Inside the plasma, not. So, outside the RBC membrane, they are present where? Inside plasma. Inside plasma. So, plasma will be containing, if the blood group is A, the antibody will be B. If the blood group is A, the antigen will be A, and the antibody is going to be B. The genotype, the last one is genotype. The genotype, we can represent it with IA, IA. That means this is homozygous. Now, what is I stands over here? I stands for isoalleles. I stands for isoalleles. So it can be homozygous A, IA, IA, or it can be heterozygous A, represented by IA, IO. IA, IO. So this is heterozygous A. Clear? So A blood group को दिखाने के लिए how many genotypes are required? To represent one phenotype called A blood group, how many genotypes are required? 2 genotypes are required. What are they? IA and or IA, IO. The second type is B. Srijan, tell me. If the blood group is B, the antigen is present over here is B only. It is on the basis of the antigen present, we can call the name of the blood group. So B blood group ki thakbe, the antigen should be B. And the antibody is going to be just opposite, A. The genotype should be IB, IB. That is homozygous B or IB, IO. So I'm writing over here IB, IO, but in your book, what is not being given? No O, only I is being given. I means what? O is present, means nothing is there. Fine. So IB-IO is a heterozygous one. But when we talk about AB blood group, that means they are having all the types of antigens. That is AB type of antigens. That is AB type of antigens. And the antibody present is no antibody is present. As because no antibody is present, we can call AB blood group as a universal recipient. Universal recipient. Is this clear? Universal recipient. Why it can receive blood from any other blood group? Because they have no antibodies. They have no antibodies. If they don't have any antibody, you can get the blood from any other what? Blood group. So that is why it's called universal recipient. Are you getting me, Ankita? Good. So how you are going to denote genotypically? It is IA and IB. It's a very, very important concept of codominance. Over here, when IA and IB, see, over here IA is dominant, over here IB is dominant. But when they are keeping themselves together, both of them are showing together AB. This was not being mentioned by Mendel, so it comes under non-Mendelian inheritance. Clear? So IA-IB acts as a codominant one. When we talk about O blood group, O means what? Nil. What is not there? What is not there? No antigen, but they have both the antibodies present. So there is no antigen. So we can call them as universal donor because they have no antigen, they can reach out to different types of blood groups. So universal donor. And the way that we can express them is IO, genotypically IO. Shrijan, tell me how many genotypes are required to express 4 phenotypes of blood group? How many genotypes are required to express these 4 of phenotypes of blood group? How many genotypes are required? 6. 1, 2, 3, 4, 5, and 6 genotypes are required to express the 4 phenotypes of the blood group. Which is to got the idea? And there is a formula. How many alleles we are talking about over here? In blood grouping, how many alleles are there? IA, IB, both of them are dominant. IO is not dominant. How many alleles are there? 3 alleles are there. So how to get the number of genotypes? There is a formula. Number of genotypes is equal to N into N plus 1 by 2. Number of genotypes is equal to N, where what is N? Where N is, come on, number of alleles. Where N is number of alleles. So how many alleles are there in blood grouping? 3 alleles. So 3 into 3 plus 1, come on, by 2. 4 into 3, 12 by 2. So how many genotypes? 6 genotypes are required to express the blood grouping. Is this clear? This is very, very important. Which is? Sure. Okay. So this is about blood grouping, but I need to tell you something more. Just wait. Yes. We're going to solve some questions, fine. But before that, let me give you some of the idea. Yes. Get this. So what we are going to cover? We are going to cover blood grouping, codominance, multiple allelism, and then we are going to start with the sex determination. Last, we are going to study about linkage and recombination. Linkage and recombination. So principles of inheritance, codominance. Both alleles express equally. Both alleles express equally. ABO blood grouping system. What is being given? See. Gene I and its 3 alleles. Gene I and its 3 alleles IA, IB, and what is I? Nothing is there means I or IO. What is IA? What is IA? It produces a specific form of sugar polymer that protrudes from the surface of the red blood cell. So they are present on the surface of the red blood cell. And what is the surface? The membrane of the RBC is called Donance membrane. So IA is going to be a particular sugar polymer. Think about IB. IB produces a slightly different form of sugar on the red blood cell surface. So the difference is IA, they form a sugar polymer, and slightly different polymer is produced by IB. What about IO? Does not produce any sugar polymer on the red blood surface. Clear? Now, when we see the diploid organisms, because humans are diploid, each person possesses any two of these three alleles. Human is diploid, so any two of these alleles will be present in a human being who is a diploid organism. Now, expression rule. IA and IB are completely dominant over small I. What is small I? IO. So who are completely dominant over the other? तीनों में से दो dominant है third के ऊपर. What is the third one? Which one is the third one? IO or I? IA is dominant over I or IO. IB is dominant over IO or I. Expression rule. IA and IB are completely dominant over I. When present together, both express their own types of sugar due to codominance. जब दोनों एक साथ रहेंगे, when IA and IB are going to be remaining together, they are going to express both of their what? Sugar polymer, and that is called codominance. That is called codominance. Mendel ki bolechilo, what Mendel said? Mendel said law of dominance. Mendel said where only one of the allele is going to be dominant over the other. Location, the gene I which controls the ABO blood grouping system is located on which chromosome number? The gene I is present, capital I is present on chromosome number 9. Autosome or sex chromosome? Chromosome number 9. Autosome है, sex chromosome है. How many pairs of chromosomes we have? How many pairs of chromosomes we have? 23 pairs. Out of 23 pairs, 22 pairs are what? Autosomes. 23 pairs are, and the last pair, 23rd pair or the last pair is what? Sex chromosome called allosome. Sex chromosome called allosome. Is this clear? Autosome, got the idea? So 22 pair में 9th pair क्या होगा? Autosome, definitely autosome. So capital I is present on 9th chromosome. That means it is an autosomal one. Understand this, fine? Okay, ABO blood group. 6 genotypes and 4 phenotypes. 6 genotypes and 4 phenotypes. So genotype क्या है? IA, blood group A, IA, A, IB, B, IB, B, IA, codominance, IO, O. 1, 2, 3, 4, 5, 6. So 6, but to express 4 phenotypes. With 3 different alleles, IA, IB, and I, there are 6 different combinations. There are 6 different combinations of these alleles. How many combinations? 6 different combinations of these alleles possible in the human population. Resulting in a total of 6 distinct genotypes. Kitna distinct genotypes? 6. An individual can only possess 2 alleles at a time. Individuals can only possess 2 alleles at a time. However, because the population exhibits more than 2 alleles for this single locus, gene locus. So how many locus are there? 1 locus for all these 3 alleles. It is a prime example of multiple allelism. It is a prime example of the same thing happens with the eye color of Drosophila. There are multiple alleles present on the same locus. The inheritance of blood groups demonstrates dominance, codominance, and multiple allelism all working together. Dominance habe? Proof how it is showing dominance. IA, IO, who is dominant? A is dominant over the other. So it is showing off law of dominance. It is showing off IA, IB is showing off codominance. Multiple allelism, definitely 3 of them are working together. Ankita, is this clear? Chalo. Next. Multiple alleles, more than 2 alleles. See, a population level phenomenon of 1 locus Multiple alleles are referred to the existence of more than 2 alternative forms of a gene at the same locus. Region, clear? At the same locus. So more than 2 alleles present on the same locus is the best example of multiple allelism. An individual carries only 2 alleles, but the population shows more than 2. This is the definition of multiple allelism. They can only be discovered through population studies since each individual carries only 2 alleles. ABO blood group inheritance demonstrates dominance, codominance, multiple allelism simultaneously, making it one of the most important diagnosis in genetics. Is this clear? See how many years the question came: 13, 20, 20 and 18. Blood grouping is a very, very important one, and we're going to solve some questions. We're going to solve some questions from blood grouping. Let us solve some questions. If the question is like this: father is homozygous towards A blood group, and mother is heterozygous B blood group, what are the chances of their offsprings having O blood group? And say the options are 0, 25, 50% and 100%. Good, very good. 0%. How? When they are saying father is homozygous A. Srijan, homozygous A means IA? Homozygous. When both the alleles are similar, we call them as homozygous allele. When both the alleles are similar, we call them as homozygous allele. And mother is heterozygous B. So IB, IO. So from here, the gamete is going to come as IA. From here, the gamete will be IB, IO. Now tell me, this is what IA, IB, 50% of the issues are going to have AB blood group. 50% of the issues are going to have heterozygous A, that is A blood group. So there are no chances of their offsprings having O blood group. So answer is, any issues with this? Got the idea? So blood grouping say questions very, very important. Acha, Saksham, tell me, name the genotypes of the parents if they have 4 different blood group in their offsprings. Tell me the genotype. एक कपल में 4 बच्चे हैं, चारों बच्चे में A, B, AB और O blood group है. Tell me the genotype of the parents. Father is going to be heterozygous A, mother heterozygous B. Then only they are going to have IAIO, and from here IBIO. If you make this Punnett square, father, mother, father IAIO, mother IBIO. So the individual is going to have IAIB, IBIO, IAIO and IOIO. So they are going to have 4 different kids having different types of blood group. That chatte bacha, 4 of them see is having AB blood group. 25% of them are having AB blood group. 25% of them is having B blood group. 25% A blood group and 25% O blood group. एनी इश्यूज? ठीक है ना? हाँ ठीक है. अगर ठीक है ये क्वेश्चन करो. ठीक है तो ये क्वेश्चंस करो. चलो okay, can we do this? Recessive epistasis, we have already done it. Recessive epistasis at F2 ratios. चलो क्वेश्चन करो. In genetic cross having recessive epistasis, what is the other term for recessive epistasis? Other term for this? Supplementary or complementary gene? Supplementary gene. So this is supplementary gene. So supplementary gene is having. Tell me the example of supplementary gene. Coat color of mice. So they have agouti is to black is to albino. Agouti is to black is to albino. We have done it already. The ratio is 9:3:4. Answer is same. Oh, answer is there. Key insights. You have seen the key insights. Good, very good. Very smart. Good. So I'm going to ask the question like this. No insight. Okay. Karo. Human skin color inheritance. Inheritance of skin color in humans is an example of which genetic principle? Skin color in humans is an example of which genetic material? It is polygenic inheritance. Very good. Polygenic inheritance. Okay. How many different kinds of gametes will be produced by a plant having the genotype? This. How many different kinds of gametes? Yes. You have to consider what? Only the heterozygous. You cannot consider the homozygous. You have to only consider the heterozygous. So heterozygous means only 1. So 2 to 1 is equal to 2. What is the answer? We saying 4. Why? Say the parent is this. What do you consider this parent as? 1, 2, 3, 4, 5, 6. Hexahybrid. You have to consider only the one which is heterozygous. 1, 2, 3. 2 to the power. The gametes you're going to have is 8. So how it happens? If the parent is like this, how many types of gametes you will be getting? Only capital A. Can it be? If the parent is like this, how many gametes you will be getting? 1, 2. 2 to the power is equal to 4. Where n is number of characters. N is number of characters. So 2 to the power n is equal to what? Diamond means 2. So you have to consider only the heterozygous one. Clear? और clear है तो करो इसको. How many gametes you can expect from this parent? No. Tell me 2 into the power? 1, 2, 3, 4, 5, 6, 7. This is the answer. If the parent is this, how many gametes from this parent? 3. Tell me the gametes. क्या होगा? क्या होगा? एक ही होगा बाबा. A, B, C. 2 to the power 0, 1. No heterozygous. Only ABC. क्या हो गया? Any problem? Tell me about this. How many gametes? 3. 2 over 3, 8. Okay? Chalo, very good. Next. करो इसको. Yes. Yes. D. Tell me how you are doing it. Incomplete dominance. Okay. Pleiotropism. Pleiotropism. Polygenic inheritance. Skin color. Pleiotropism we are going to study today. Pleiotropism. What is pleiotropism? One gene, one character, or one gene is expressing when one gene expresses more than one character. When one gene expresses more than one character, we call it pleiotropism. So, what is codominance? Blood group. What is incomplete dominance? Flower color in antennatum. What is pleiotropism? Phenylketonuria. What is the answer then? B is the answer. B is the answer. Okay. Okay, go for this. Matching terms and descriptions. Karo. Matching terms and descriptions. What is dominance? Tell me. A single gene expresses many characters. Pleiotropism. A single gene influences many characters. Pleiotropism. Codominance. In a heterozygous organism, both alleles express themselves fully. Dominance. In a heterozygous, only one allele expresses themselves. Come on. Kire. Eta ki hobe? IA? IO. Eta ki? IA? IB. Ye hoga. Polygenic. Many genes govern a single character. What is the example? Skin color. क्या था ये? Negro. Completely black. Negro. ठीक है? What is this? Skin color. Albino. What is this? Skin color. Mulatto. Intermediate brown. Clear? Answer क्या होगा ये? Principles of test cross. Do you know what is test cross? What is test cross? What is test cross? Capital T cross with small t. This is P1 generation. Parental 1. From here you will be getting gametes capital T. From here small t. So this is gamete. This is F1 generation. You will be getting capital T small t. This is heterozygous tall. If you make a cross between F1 hybrid with the recessive parent, this is called test cross. And the ratio is always going to be coming as 1:1 only. Test cross में ratio क्या आएगा? Cross F1 hybrid with recessive parent. Recessive parent. So capital T small t cross with small t small t. क्या होगा? From here, you will be getting capital T and small t. From here, small t small t. So we're getting capital T small t and small t small t. So what is your tall is to dwarf. Ratio 1:1 always. So in test cross, the ratio is always going to be 1:1. Clear? बोलो समझ में आया कि what is test cross? So tell me what is back cross? A cross we are making with either this or this. It's called back cross. So what we can say? All test cross are back cross, but all back cross are not. All test cross are back cross. So what is back cross? Shuman, বুঝেছিস? What is back cross? F1 hybrid এর সাথে dominant parent or recessive parent যে কোনো একটা হলেই এটা back cross হবে. And what is test cross? Cross between F1 hybrid with its only বুঝেছিস? তাহলে all test cross are back cross, but all back cross are not. Sujan, clear? Sure. Good. Give that answer. What is the answer? Crossing the F1 hybrid with a double recessive. What is double recessive? Small t is double recessive. Chalo, the one which we are studying. Blood group inheritance and codominance. If two persons with AB blood group marry and have a large number of children two persons with AB blood group marry and have a large number of children, they could be classified as ABB in a ratio 1:2:1. Modern protein electrophoresis reveals the presence of both A and B types in AB individuals. This is an example of why in AB individual they are having A and B protein. Remember, A क्या देता है? What A provides? It gives rise to a sugar polymer. And what about B? Different type of polymer. So both of them are giving rise to what? Proteins. So answer क्या होगा? Codominance नहीं होगा. And should be codominance. चलो ये बहुत इंपॉर्टेंट है, बहुत टफ है, करो. Shuman bol. A mother in a family of 5 daughters is expecting her 6th baby. The chance of its being a son is Durba. A mother in a family of 5 daughters is expecting her 6th baby. The chance of its being a son क्या होगा? अच्छा 0 क्यों? 6 हो गया इसलिए 5 हो गया इसलिए लड़की ही होना है. What is the science? What is the science? हाँ 50-50 is the science. क्यों? नहीं साइंस क्या है? बोलो 50-50 कैसे बोला? सेक्स डिटर्मिनेशन सोचो. फादर क्या है? XY. मदर क्या है? XX. So father can give rise to 2 types of sperm. One of the sperm containing X chromosome. Father is heterogamete. And the sperm, it can have Y chromosomes. But mother can always give rise to an ovum having X chromosome. So mother के ऊपर तो है ही नहीं ये बात. क्लास 10 पढ़ लिए थे आपने. Mother के पास अगर ये sperm आता है, what is the sperm called? Sperm containing X chromosome. কি বলে এটাকে? Gynosperm. The sperm containing X chromosome is called gynosperm. The sperm containing Y chromosome is called androsperm. क्या कहते हैं इसको? Androsperm. If gynosperm fertilizes this ovum, Shuman কি হবে? ছেলে নামে XX কি মেয়ে না ছেলে, so they're going to have daughter. If androsperm fertilizes this ovum, they're going to have XY, son. So what is the chance of getting son? 50%.
सर तो मतलब कि हर बार मतलब कि 6 हुआ अगर 7 हुआ तब भी 50%.
पहले में भी 50% था, सेकंड में भी 50% था, थर्ड में भी 50% था.
50% था है रहेगा.
क्यों? साइंस तो यही बोल रहा है. Because nobody can say if this है ना? इन दोनों में यहाँ पे आने का चांस कितना परसेंट है? 50%। दोनों में से एक जाएगा तो व्हाट इज द चांस? 50%। इज दिस क्लियर? यहाँ पे 6 में भी ना होकर अगर 10th में भी होता तो क्या होता? क्या बोल रहे हो? 100/10 कितना होता है? 10%। यहाँ ऑप्शन नहीं है, तो क्या है आंसर? क्या होगा? अच्छा 50%। ओके चलो ठीक है। पॉइंट म्यूटेशन पढ़ा ही नहीं हमें, वी हैव नॉट टॉक्ड अबाउट। सो इनहेरिटेंस समरी, ओके वेट, क्लियर? चलो yes, let's come into the whiteboard. So this was about blood grouping. बोलो समझ में आया? Blood grouping is clear. Blood grouping में कोई डाउट नहीं बाबा? हाँ अंकिता, so you can solve any of the questions from blood grouping. So blood grouping is very, very important. It's going to come in your exam. Okay, very good. चलो next. Let's get to the next term, and next one is called pleiotropism. क्या नाम है? Pleiotropism. The next one is about pleiotropism. What is pleiotropism? When one gene, normally what happens? One gene expresses for only one character. One gene expresses for one character. But when one gene is going to express for more than one character, then this will be called as pleiotropic gene. क्या नाम होगा? ये जीन को क्या कहेंगे? This one will be called as pleiotropic gene. Pleiotropic gene. The bestest example of this is sickle cell anemia. देखो यहाँ पे बुक में देखो, sickle cell anemia. Sickle cell anemia. What happens to the RBC membrane? Normally RBC remains like this. This is the structure of RBC. Srijan, what is this particular structure called? Biconcave. So the normal RBC, they have got biconcave shape. So this is biconcave shape. But if the RBC becomes sickle shaped, the amount of hemoglobin will be less or more? Less. Less in this, but more in this. Clear? Hemoglobin ज़्यादा रहेगा biconcave shape होने से. But if the RBC shape becomes sickle shaped, definitely they are going to suffer from anemia. Lack of hemoglobin will be called as anemia. But ये sickle cell anemia, ये एक जीन के लिए होता है. If there is a problem with one particular gene and it becomes pleiotropic, then what can happen? Then sickle cell anemia can occur. This, we can also call this one as a point mutation. Point mutation. Do one thing, check out in your book. Next day we are going to go in depth of it. यही फिगर दिखाएंगे आपको. 6th पोजीशन में देखो. Normally we know that hemoglobin. What is hemoglobin? In class 11 we have studied about hemoglobin. Tell me. It consists of what? Heme plus globin. What is heme? Heme is iron. What is globin? It's a protein. Varun, tell me, what kind of protein is this? We have classified in the chapter biomolecules, we have classified protein into different types. One is simple protein, conjugate protein, and derived protein. It comes under conjugate protein. What is conjugate protein? When a protein is not only made up of amino acids, but along with that, it is also made up of some prosthetic groups. क्या नाम है? It's the best example of what? Either metalloprotein. So what is the metal out here? Iron, metalloprotein, or we can also call it as pigments are present. Chromoprotein. What is the color? Red. Now, protein is made up of 2 alpha chain. Globin protein is made up of 2 alpha chain and 2 beta chain. 2 alpha and 2 beta chain. Where each alpha chain contains 141 amino acids. Each alpha chain contains 141 amino acids. Each beta chain contains 146 amino acids. In the beta chain, especially. So we are going to study about alpha thalassemia, beta thalassemia. Thalassemia is there in your book. Fine. Alpha thalassemia, what happens? There is some change, some problem in the alpha chain of the globin protein. What happens in the beta thalassemia? There is some problem in the beta chain of the amino acid. Beta chain of amino acids of the globin protein. Now, what happens in sickle cell anemia in the beta chain? You can see in your book, there are 6 amino acid being given, shown over there. 1, 2, 3rd, 4th, 5th, and 6th. In the 6th one, it is the normal RBC, the normal hemoglobin, the normal RBC where the normal hemoglobin is present. See what happened? What is present over there in the 6th position? Glutamic acid. Even in the 2nd, 7th, what is present? Glutamic acid, GA. GA stands for what? Glutamic acid. But this protein chain, this is amino acid. So many of the amino acid together makes what? A protein chain. Tell me the bond present between the 2 amino acid. Peptide bond. So what is a protein made up of? It's made up of polypeptide chain of amino acid. It is made up of polypeptide chain of amino acids. So many of the amino acids together, they make a bond, and the bond is called peptide bond. याद है ना कैसे बनता है? पानी निकल जाता है. Remember, we have taught this in class 11. Carbon is having 4 arms. At one arm, it is having COOH. Other arm, it is having NH2, alkyl group in the other arm, and hydrogen. So this is the structure of amino acid. So 2 of the amino acid. Which one is the variable one? Alkyl group. Do you remember? If the R is replaced by hydrogen, we call it the simplest most amino acid called glycine. If this R is replaced by hydrogen, we call this amino acid as glycine. Say there is another amino acid. So this is glycine. Say there is another amino acid and the R is replaced by methyl group CH3. And this is NH2. We can write like this. This is hydrogen and this is COOH. So an amino acid is having amino group and carboxylic group. So when 2 of the amino acids are close together, यहाँ से hydrogen निकल जाएगा, यहाँ से OH निकल जाएगा. So what goes out? H2O goes out. Srijan, की रोएगा लो? CO, NH. क्या रह गया? CO, NH. So what is this bond? CO, NH. What is this bond? CO, NH. This is a peptide bond. So think about it. In the 6th position of the amino acid, what is present? Glutamic acid is the amino acid. Say. And this glutamic acid is represented by GAG as mRNA. Normally हम जानते हैं mRNA से क्या बनता है? Protein. The process is called as transcription. The process is called as transcription. So from mRNA, the protein is formed by the process of transcription. And mRNA is a single stranded. mRNA is a single stranded. So it's a double strand. 5 prime to 3 prime, 5 prime to 3 prime. This is ATGC. This is TACG. ये क्या है? Double bond, triple bond, triple bond. जानते हो ना? जानिश ना? T is purine, T is pyrimidine, and then adenine binds with thymine with a bond. Guanine binds with cytosine with a triple bond. So this is DNA. What is this? DNA. From this double-stranded DNA, the DNA makes what? mRNA makes protein by the process of translation. Translation. So next chapter we are going to study about all this. So DNA is going to make mRNA, single-strand mRNA. This is single-strand mRNA, starting from 5 prime to 3 prime. And the process is called as transcription. The process is called as transcription. But in this mRNA, this TACG, T से क्या बनेगा? A से क्या बनेगा? U. Because in mRNA, adenine binds with uracil and cytosine binds with guanine with a triple bond. So in place of A, it will be U. In place of C, it's going to be G. And in place of G, it's going to be C. Clear? So if this is so, वहां पे देखो, in this GAG, it has come from what? इसके ऊपर mRNA में क्या होगा? बोलो, अगर GAG है, GAG, it is an mRNA, 5 prime to 3 prime. बोलो, DNA में क्या होने वाला है? In case of DNA, Durva, tell me. This is 3 prime, 5 prime, and this is 5 prime, 3 prime. Tell me, क्या होगा यहाँ पे? G कहां से आया? From C. A कहां से आया? From T. G कहां से आया? From C. इसका ऑपोजिट क्या होगा? GAG। बोलो समझ में आया कि नहीं आया? सो इन डीएनए से क्या बनेगा? ये mRNA की तोरी हलो mRNA। इन द सिक्स्थ पोजीशन दिस गैग इज गोइंग टू रिप्रेजेंट glutamic acid normally. But what has happened? अब देखो वहां पे आरबीसी का स्ट्रक्चर नॉर्मल है, बाइकॉन्केव है, बट व्हाट हैज हैपेंड? ड्यू टू अ पॉइंट म्यूटेशन दिस गैग हैज बीन रिप्लेस्ड बाय गुग। द गैग हैज बीन रिप्लेस्ड बाय गुग, so this is called point mutation. Gag has been replaced by gug, and gug is going to represent what? Glutamic acid or valine? Valine. So glutamic acid is replaced by valine. बोलो समझ में आया क्या? Glutamic acid in the sixth position, glutamic acid has been replaced by valine. And due to this point mutation, the whole of the one gene has been changed, and this gene is having expression of more than one character. And what is the character? The RBC shape becomes this, sickle shaped, and the amount of hemoglobin is reduced. And this is called sickle cell anemia. We will talk about that later. Next day we are also going to talk about this. बहुत clear na. So sickle cell anemia is an example of pleiotropism. Under pleiotropism, the next example is PKU, phenylketonuria. We're going to study after that. See Mendelian disorders. We're going to talk about that. Fine. But today, I need to talk about sex determination in animals. In your book, it's very, very important. When we talk about sex determination, one gene, one character is okay. Mendelian concept. What about one gene, more than one character? It also comes under non-Mendelian inheritance. So pleiotropism is also an example of non-Mendelian inheritance. Ball it clear. Kono অসুবিধা আছে? Shaksham clear? Srijan, tell me what we are going to study? Sex determination. See, we need to study about 4 organisms. 1, 2, 3, and 4. The first one about human. Second one about birds. Third one about the second one is Drosophila, that is fruit fly. The third one is birds, and the fourth one is bee. There are 4 examples being given in your book. The first one you already know. What is the first one? The first one is human. Chal dekheni, human, I think you know it. Just now we were talking about this. Human. Srijan, how many chromosomes? 44 plus. This is father. Having 44 means 22 pairs. And the last pair is sex chromosome, XY, allosome. Cross with female, 44 plus XX. This is mother. So from father, we can get 2 types of sperm. One having 22 plus X, and another sperm containing, Sumon, ball, 22 plus Y. 22 plus Y. This has been a complete disjunction. यहाँ की वजह से देख, यहाँ पे क्या बन गया? Complete disjunction of chromosomes has occurred. Why I'm telling you? Because in later part of our class, we are going to talk about non-disjunction also. We're also going to study about non-disjunction also. Shumon, বুঝতে পারছিস? From mother, she is only going to give rise to what? 22 plus? The X will be having 22 plus X. So if, Srijan, tell me, if this particular sperm, this sperm is what? Gynosperm. The sperm containing X chromosome is called gynosperm. The sperm containing Y chromosome is called androsperm. So if this gynosperm fertilizes this X, we're going to have 44 plus XX. Daughter, definitely daughter. So they are going to give rise to daughter. Saksham, if this androsperm fertilizes this X, we're going to get what? 44 plus XY, so son. So tell me, यहाँ पे heterogamete कौन है? Father या mother? कौन दो different types of gametes बना रहा है? Father या mother? So we call it male heterogamety. Outline the term in your book. Male heterogamety. So they give rise to male heterogamety. So the male is, the human male is heterogamete. Srijan, tell me why? Why the human is called as a heterogamete? Why the human is called as a heterogamete? কেন বলছে? কেন বলছে বল? Why the human is called heterogamete? Because they are having two different types of gamete. See, the male is having দুটো type of gamete আছে না? একটা sperm having X chromosome, sperm containing Y chromosome. Clear? So male heterogamety is shown by human. Clear? Let's talk about Drosophila. In Drosophila, the same thing happens. In Drosophila also, the male is XY and the female is XX. So what is the chromosome number of Drosophila? Who is going to tell me? What is the chromosome number of Drosophila? Any idea? 2n is equal to 8. So what is n? Is equal to 4. N is equal to 4. So 6 plus XY male cross with female 6 plus XX. Sijon ball, how many gametes from this father, from this parent, male parent? 3 plus X and the sperm containing 3 plus Y from this female. They're going to only have 3 plus X. Is this clear? यहाँ पे male heterogamete है या female? Who is heterogamete? Male or female? Sijon. এখানে মেল দুটো গ্যামেট দিচ্ছে হেটেরো মানে কি রে ডিফারেন্ট हेटेरो मतलब क्या है डिफरेंट सो डिफरेंट गैमेट कौन दे रहा है सर मेल is giving different types of gametes so this it also shows the male heterogamety clear just like human see in human also the male was heterogamete great in birds the male one is whatever chromosome they have, ZZ. Cross with female is ZW. So who is going to give rise to two different types of gamete? Female is going to have different types of gamete. One will be containing Z and one will be containing W. So two eggs having two different types of chromosomes. So यहाँ पे कौन है? But male is only going to have Z in their sperm. So one type of gamete. So ये male homogamete है, but female क्या है यहाँ पे? What is female over here? Female is heterogamete. Female is heterogamete over here. It's called as female heterogamety. देखें ना वो ये लिखा है. Just have a look. Female heterogamety कहाँ पे लिखा है? Female heterogamety. Clear? Female heterogamety has been given. Clear? Female heterogamety. Come down to bees. These bees we are talking about drone. Drone means male or female? Male. Mind you, male are always having haploid number of chromosomes. But female, they have diploid organisms. Not only female. One is female which is called as workers. They are female having 2n. And 2n is equal to 32. 2n is equal to 32 in B. And in the other one is called queen. Queen is also female. Queen is also having diploid. So 2n is equal to 32. So all the males, they are drones and they are haploid. So haploid कैसे बन रहा है देखो. And the concept of this determination is called diploid sex determination. And what happens over here is, say this is male having n number of chromosome and n is what? 16. And this is female having 2n is equal to 32. So यहाँ पे क्या होगा? Meiosis होने वाला है. What is going to happen over here? 2-1 is going to show meiosis. So there will be two types of ovum or two types of eggs having 16 and 16. That is N and. But because male is haploid, you cannot expect meiosis happening. We can expect mitosis happening. And it's going to give rise to a gamete which is having 16. So if this 16 from male fertilizes this female ovum, egg, it's going to give rise to what? 32. So 2n is equal to 32 means it's female or male. And if this ovum remains unfertilized, there is no sperm to fertilize it, it already becomes what? N means what? Male. So this is the male honeybee. Male bee which is haploid. And this particular life has been formed without fertilization. This is an also example of parthenogenesis. It is also an example of parthenogenesis. What is parthenogenesis? What is parthenocarpy? Formation of fruit without fertilization. And what is parthenogenesis? Formation of life without fertilization is called parthenogenesis. Is this clear? So this is called as haplodiploid type of sex determination. That's all for today. Next day we are going to start with pedigree analysis, and we are going to start with, to understand pedigree analysis, we need to study about Mendelian disorders. So Mendelian disorders means some of the disorders are there in autosomes, some of the disorders are there in sex chromosomes. So we are going to talk about autosomal traits and autosomal disorder and sex-linked disorders. Okay, that's it for now. Chalo, aaj ke ruk tha. Chalo.